Okay, good morning everyone. Welcome to the last class of CS 206. This will not be the last time we meet. Remember, next Tuesday morning, bright and early, 7:30, we will be here for the final project presentations. A couple of housekeeping things before we get started. I finally remembered to bring in one of the soft voxels that makes up the soft robot, so feel free to have a look and pass it along. Okay, something to play with while we get through the housekeeping materials here. A/B testing. Before we get to A/B testing, student evaluations, please fill out the course evaluations. I know you're super busy. I know there's a million things to do before the end of the semester. I look at the course evaluations. Please be honest, what worked, what didn't work. I try and incorporate it into next year's class. So don't help me, help the students that are going to take this class next year. Please fill out the student evaluations. Very much appreciated. Okay, I know I've mentioned A/B testing quite a few times. I just want to go over this one more time, suggest a few more details to help with A/B testing. You are trying to answer a question decisively one way or the other. You have between now and Tuesday morning to convince yourself, and then Tuesday morning you're going to try and convince myself and your 59 other student colleagues that either your A is better than B is better than A is better than B is better than A, or A and B are more or less equivalent, meaning there's no real, B didn't really help or hurt your evolutionary algorithm to find a solution for your robot, right? Okay, so we have evolutionary time on the number of generations on the horizontal axis. We have fitness on the vertical axis. For some of you, lower fitness is better. For others, larger fitness is better. Let's assume that higher fitness is better. You get 4 runs of A, and then you get 4 runs of B. And there's no appreciable difference, so you move on to a new A and B. Are you convinced that this is the answer given this graph? Is 4 runs of each enough, right? I see some of you kind of wagging your heads. It's not enough. There is always a temptation in science to jump to the next hypothesis, right? Okay, there doesn't seem to be much of a difference. Let's change something else. For some of you, you may only be performing 10 generations, so you go back and start up A and B again with 100 generations. And between now and Friday, you get a couple of curves of A and a couple of curves of B with 100 generations. And Tuesday morning, you bring this plot and then a separate plot that has 2 A curves and 2 A curves for 100 generations, which also isn't going to be enough to convince us one way or the other, right? So my suggestion is resist the urge to move ahead to yet another modification to A and B. Whatever time you have left between now and Tuesday morning, get as much data as you can from your current A and your current B. And if you get 10 curves of A, 10 curves of B, 20 of A, 20 of B, and there's still no difference, at least we're going to believe you more that there is no difference between A and B. Clear? Okay, so for the computer scientists in the room, pursue depth first rather than breadth first. Collect as much data as you can with your current version of A and B. The more data you have, the more you're going to be able to convince yourself and others which of these 3 questions is true in your current setup. Mentioned last time that some of you are more comfortable with statistics than others. If you want to plot means and error bars and p-values, that's perfectly fine. If you're not comfortable with statistics, that's fine too. Data is your friend. The more data you collect, the less fancy statistics you need to do to try and convince us which of these 3 is true. Okay. That's all I have to say about A/B testing. Anyone have any final questions? No? All good? Okay, so as promised, I just want to walk through again what's going to happen on Tuesday morning so we're all on the same page. We are going to get through 60 2.5-minute YouTube videos in less than 3 hours. you must submit your final project materials to us by 11:59 p.m. because we need to stitch all those 60 YouTube videos together into a playlist. If you submit a link and it's broken, or it's a private link, or it's 10 minutes long rather than 2.5 minutes long, it's out. We're not putting it into the playlist, and you will not be able to present your work orally on Tuesday morning. Yeah? So make sure it's pretty much 2.5 minutes. 2 minutes and 29 seconds, 2 minutes and 31 seconds, that's fine, but really try and keep it as close to 2.5 minutes as possible. Okay. We will stitch all of these into a playlist. I will be up here at the front, as always, running the YouTube playlist. I will write up on the board, when you come in on Tuesday morning, there will be 60 names on the board of exactly when you're presenting. So when the person before you is presenting, please come up to the front of the room on the far side of the screen there to be ready. When you present and you're done, come on back around this way, sit down, because the next video will immediately start and the person on deck will immediately start speaking. We are not going to stop and start the playlist 59 times between the 60 presentations. We'll be here much longer than 3 hours. Make sense? Okay. Last thing, we have a lot of people out sick for obvious reasons. If you are ill and you are not able to present, please let me know as soon as you can. If you are sick and you are not able to physically come to class and physically present, there is a plan B, which is the following. You still record your 2.5-minute video, but I want you to put captions on your video in sufficiently large font so that students in the back row would be able to read your captions. Yeah? You can submit a video with captions if you're ill and send me a note that you cannot come in physically to class, and we will stitch your video into the playlist as usual. It will obviously just run silently, and we will all read your captions. You can't narrate your voice over the video because I am not smart enough to figure out how to get the audio to work at the front of the class here. So we're going to do everything with captions. Yeah? Okay. Any questions about that? If you do find that you're ill and you need to put captions on your video, as I mentioned, please put it in large font. Do not copy and paste paragraphs into each of your slides. There's no way we're going to be able to read it fast enough to keep up. So if you are adding captions, add captions, and then play your video and read as your video plays. If you can't keep up with your captions, neither can we. Any questions about the logistics of how this is going to run? All good? Okay. All right. So we are finishing this final theme of the course on expanding evolutionary algorithms to optimize as many aspects of the robot or the biobot as possible. We are going to finish lecture 26 over the next 20 minutes or so. We're going to see how we actually go about making Xenobots once they've been evolved in silico, and how and why they cross the sim-to-real gap. When we finish lecture 26, before moving on to lecture 27, we're going to pause and I will just open the floor. I have done all the talking in this class. We have covered a lot of material. We have not talked about the ethics of robotics or biobotics, so we'll open it up for general discussion in about 20 minutes' time. Any sort of questions that remain in the back of your mind that have nagged you about various topics we've talked about, or other topics you have questions about, we'll have an open discussion. If that open discussion takes us to the end of class, that's perfectly fine. You can go read about self-replicating Xenobots on your own. If our open discussion winds up before the end of class, I'll at least describe in broad brushstrokes how Xenobots self-replicate. Sound like a plan? Okay, so back to lecture 26. We were looking at Xenobots last time. I told you a little bit about the origin story. We showed our biology colleagues at Tufts, our simulated soft robots. They built a frog sculpture, a biosculpture out of frog skin cells, which doesn't move. I explained how we were transitioning from building a biobot or a biosculpture to a biorobot by incorporating or extracting two types of cells from very early frog embryo, basically frog egg, frog skin cells, and frog heart cells. Heart cells increase and decrease in volume spontaneously. If you combine frog heart tissue together, as far as we know, if you combine it together in a shape other than a frog heart, those patches of heart tissue do not synchronize. You can see the red-green voxels here, which are simulated frog heart tissue. At some points, some of the voxels are increasing, and at the same time, some of the other voxels are decreasing in volume. We introduced a very old idea from sim to real going back to the mid-90s, which is if you're not sure about certain details of the real world or the real robot, which in this case is whether or not heart tissue will synchronize, be conservative. If you're either sprinkle noise, as we saw in the radical envelope of noise hypothesis, or in this case, just do not allow any of the simulated heart tissue to synchronize. This makes things particularly difficult for the evolutionary algorithm. It's got to figure out how to combine together unreliable parts that are increasing and decreasing out of phase with one another to produce a reliable machine, a reliable simulated Xenobot that moves as forward as quickly as possible. We wouldn't be here talking about this if it turns out that it wasn't possible for the evolutionary algorithm. It is, and we'll see how in a moment. I mentioned last time how the first attempt at this, not surprisingly, the evolutionary algorithm, when we did 100 distinct evolutionary trials, we got 100 balls of muscle at the end, which Doug, the microsurgeon at Tufts, told us there's no way he could make such a biobot. And even if he could, it wasn't going to jump along the bottom of the Petri dish. Frog heart muscle tissue is not that strong. So we went back, we did real to sim. We took the information that the biologist gave us and put it in the simulator. We changed how these voxels are simulated so that the red-green voxels, the heart muscle, is weaker. And it's not allowed to use too much heart muscle tissue. It had to incorporate the passive skin tissue as well. Okay. So after that, we did another 100 evolutionary trials and got these 100 champions back. These are the most fit robots from each of the 100 independent trials. Crossed our fingers, sent this to our microsurgeon colleague at Tufts and asked him whether he could build any of these 100. And the answer was yes. 5 out of these 100 were buildable. We asked Doug, how do you know that those 5 are buildable? And after a discussion back and forth, we boiled that discussion down into a flow chart, which is take a given one of these designs and start at the top of this flow chart and ask the following question. Does that Xenobot have contiguous tissue regions? Meaning, if you have a heart voxel, then a skin voxel, then a heart voxel, then a skin voxel. If you basically have a checkerboard of skin and heart, you don't have any contiguous pieces of tissue. Doug said there's no way I can put one heart cell and then one skin cell and then one heart cell and then one, I can't do it, it's impossible. So if the answer to this question is no, there are no contiguous regions, then that design is out. We've already failed to cross the sim to real gap for that design. If the answer is yes, there's a potential that that design might cross the reality gap. So let's go on to the next question. The next question is it a stable geometry? What do we mean by a stable geometry? A stable geometry is once Doug builds the Xenobot, you're going to see this in a moment, it doesn't necessarily hold still, right? If you build a robot out of dead metal and ceramics and plastics, it's not going to change shape on its own. But if you build a machine out of cells, the cells talk to one another, and they have ideas about what they want to do, which might not agree with what you want them to do. So even if Doug is able to build one of these Xenobots, it's not necessarily going to hold the shape. It's not going to be a stable geometry. The cells might change the shape. What we found so far is if the Xenobot has a small concavity in it. So it may be difficult to see. Let me break out of this and zoom in a little bit so you can see better. You'll notice that some of these designs have little concavities in them, little inward pointing curvatures. Here's a couple that are, or here's 3 that are actually hollow. It turns out that the cells, for whatever reason, don't like small concavities. If they see one, they will close it up. If the concavity is large enough, it's like 1/3 or 1/2 of the diameter of the Xenobot, the cells will hold that concavity for whatever reason. A lot of what's going on in the Xenobots here is unknown at the moment. We are still learning about sort of what works and what doesn't work. So as long as there aren't overly small concavities in the shape of that Xenobot, we can move on. It's still possible to create that Xenobot. If there are too small, concavities, Doug might be able to build that Xenobot, but it's not going to hold the shape, so don't bother building it. We failed to cross the reality gap. So far, so good? Questions? Okay, if a given Xenobot design passes these 2 filters, we move on to the 3rd one, which is that the design needs to have at least 50% passive tissue, 50% skin tissue. This seems like kind of an arbitrary choice. The reason is, again, we're trying to guard against balls of muscle, but we also want to leave room, so to speak, for other things. At the moment, we can make Xenobots out of skin and heart tissue. We're already at work on making biobots that can be built from other cell types. So we're kind of leaving room in these designs to add additional cell types or additional biological components into these designs. What do you think some of those biological components might be? What are these admittedly simple robots missing? Neurons. There are no neurons yet. Yeah. So neurons is definitely near the top of the list. What else is missing? Sense organs or sensory cells, absolutely, right? So we've talked about open loop and closed loop control in this class. Open loop control is that the motors just do something over and over again because the robot is blind. It can't sense its environment. The current Xenobots that you're going to see, these are open loop control. The heart muscle just does what the heart muscle is going to do. It's not influenced by the environment in any way. So we worked on leaving room in these biobots, and we're now working on incorporating sensory cells and neurons to try and close the loop. Can we create a Xenobot that senses its environment? That sensory information passes through the neurons inside the Xenobot and influences the muscle tissue. And the actuators would then move the Xenobot, which would change what the Xenobot senses, and we have closed loop control. We have a Xenobot that pushes against its environment and is able to sense the repercussions of its actions, right? We're not quite there yet, but we're paving the way for that. Okay. It turns out that for all of these, for 95 of these 100 designs, they get knocked out at this stage, or this stage, but 5 out of the 100 made it down here. That before we built them, it seemed that it would be possible that we could build those 5, and if we built them, they might exhibit what the evolutionary algorithm evolved them to do. Okay, here's one of the 5 that passed the test. Here it is in the VoxCAD simulator. Here's a Xenobot with more or less randomly behaving actuators, the muscle tissue on the bottom, and we've got the passive skin material on the top. And we should have drawn the trajectory, but you can sort of infer it by watching this video that the Xenobot moves more or less in a straight line from left to right. How did the evolutionary algorithm solve this problem? How did it create a reliable machine out of unreliable parts? There's several hints in the video itself that have to do with the 3D geometry, material properties.
By being bilateral symmetric.
It's bilaterally symmetric, so that's always a good sign. Yeah.
It seems like each neuron is set to fire off at a set time. Like each interval is exactly the same. There's no variation between each neuron or muscle fire.
Every muscle fiber, right? So each of the red-green voxels is a small patch of muscle tissue. They all have the same frequency. You can see that they're all turning red and green at about the same rate. Doug told us that that's probably a reasonable estimate. But the phase offsets are randomly set different to one another, right? One of them turns red, each of them turns red at a different point in time. The amplitudes are all also the same, meaning all the red-green voxels get to the same maximum size and decrease to the same minimum size or volume also. But how? How is it channeling all of that into reliable forward motion?
Stable and moving forward.
It's stable, right? So it's relatively low to the ground. It doesn't have big, tall legs. So evolutionary algorithm has struck a particular trade-off between stability and motion, stay relatively low to the ground. And it's also tipped forward. So the mass distribution is tilted a little bit forward, which helps. Why does it help? Given your experience with physics engines now, and all we've talked about physics engines, what is it about the physics in this physics engine that's helping something that tips forward to move forward? Friction, absolutely. So I mentioned last time that early frog cells are sticky. We're going to allow them to walk along the bottom of a petri dish, which is also relatively sticky, which means in the physics engine we built in relatively high friction between the voxels and the ground. So if your quote-unquote legs are pointing down and behind you, and you're pistoning those legs into the ground, generally speaking, that's going to move you forward. But that doesn't guarantee that you're going to move forward in a straight line. You could move forward in a random, tortuous path. What's the additional detail here that's allowing the robot to move relatively straight? Bilateral symmetry helps. Somebody mentioned that already. So one additional detail that the evolutionary algorithm has hit on. If you look carefully at the blue voxels on the top, this is passive soft material. These are skin cells that are not applying forces. They are absorbing forces. You'll kind of see that all the random impulse forces coming from the ventral surface or the bottom part of the Xenobot are kind of pushing up into the blue voxels. And those voxels, the blue voxels together, are kind of absorbing or cushioning all those forces and directing it forward. So in a way, the skin cells seem to be sort of averaging out the randomness or the random phase offsets in the forces coming from below, and somehow that's helping it move forward. I would be more precise about how exactly that works, but we're still not sure. It's kind of a new phenomenon here. Again, it's another useful feature of building from soft materials. Question?
Did you find that when you built them, the skins or the muscle cells actually did go in random phase offsets?
Great question. So when we built this, do the muscle tissue, patches of muscle tissue also move in random phase offset? I'm going to show you that Xenobot in a moment, and you can judge for yourselves. Okay, so here we go. This is our design. We're going to try, Doug is going to try and build this from frog cells to see whether this particular design crosses the sim to real gap. We're going to take a few minutes, and I'm going to tell you some of the details about how these Xenobots are built. This is not a cell biology class, so we're going to skate over a lot of biological details here, but I'll give you the basic idea. What you're going to see is a series of videos taken through the microscope while Doug is trying to build a Xenobot. These videos were taken not from him building the Xenobot I just showed you, but building the original Xenosculpture here. Okay, here we go. So what you're seeing here, these are fertilized early frog embryos. As you can see, they're basically frog eggs. you're going to see, oops, I skipped one here. He's injecting a small amount of reporter protein, which is going to light up, and it's going to allow Doug to identify the different cell types that he's after. He's going to try and extract precursor skin cells. So these are stem cells that are going to become skin cells, and also heart muscle tissue. There's a whole bunch of other cell types, obviously, in these early embryos we're going to try and ignore. So this is Doug preparing to be able to see what he's going to go after. Each of these early frog embryos is covered with a vitelline membrane, which is something we don't need. You'll notice Doug going in with microforceps. Remember that each Xenobot is about 1 millimeter across. So this is like trying to manipulate a speck of pepper with very, very, very small instruments. You can see even for an extremely accomplished microsurgeon like Doug, it takes a lot of effort to get this just right. Okay, so kind of removing the unwanted material. Next task is to remove animal caps. The animal cap is the cap or the top part of the early frog embryo, which tends to contain most of the cell types that we're interested in. The lower part that we're going to throw away is mostly egg yolk and some other stuff that's not needed. So we're extracting just the part of the embryo that's needed. So far, so good. Any questions? Okay, so there's two animal caps on the left now. These contain the cell types that we're interested in. Now you can see these 1, 2, 3, 4, 5, 6 animal caps again. And in this case, now Doug is removing the ectoderm from dissociated inner skin cells. So you might remember from Anatomy 101, skin is made up of different layers. We want to get rid of the ectoderm, the outer layer of skin, and we're trying to get at the innermost layer of skin. And you can see that now as these very, very small white spheres. Each individual white sphere here is one frog skin cell. This is the raw material that Doug is going to use to make the sculpture on the left. So far, so good? Question?
Is there a computer that he's somehow controlling the forceps, or is he using it with his hands?
There is no computer controlling these forceps. This is artisanal biobotics. Every single Xenobot to date has been built by hand. Design is automated with the evolutionary algorithm, but fabrication is still completely manual. Takes Doug about 4 hours to make 1 millimeter sized Xenobot. These are extremely expensive machines in terms of human time and effort. Okay. Once Doug has got rid of the ectoderm, he's drawn off with a very, very small syringe these innermost skin cells, and you can see him now pushing these skin cells out of this very small syringe into this dark circle. This dark circle is a very small well or a small concavity in the bottom of the dish, and these cells are going to settle. They're going to settle into this well in preparation for sculpting. I think this is more of the same. You can see multiple wells here. So usually, obviously, since it takes so long to make a Xenobot, it takes a lot of effort to extract this raw material. He's going to inject these cells into multiple wells and make multiple Xenobots in parallel. Once they're in the well, Doug leaves them there for a few hours, and something pretty amazing starts to happen. I'll leave this on loop. What's happening? Remember that each sphere that you see here is an individual frog skin cell. What's happening?
They like reattach together.
They reattach, right? Humans are very social creatures. Turns out that cells are as well. They don't like to be on their own. They will try and readhere as best they can. Now, how actively they are trying to seek out other neighbors and grab a hold, that's still hard to say. These cells are immersed in room temperature pond water. And at this scale, there are very small microcurrents in the water itself, which are kind of buffeting the cells around. The cells are very sticky. So if you imagine you balled up a whole bunch of balls of tape, and then you suspended them in liquid and sort of agitated that liquid, they'd bounce around, and if they bump into one another, they'd adhere. But you can also notice there's definitely some pulling together. Even after they readhere, they start to densify or pull into a ball. My biology colleagues refer to this as rebooting multicellularity. We've taken a multicellular creature, an early frog embryo, pulled it apart into its component pieces, and it seems like these component pieces want to come back together and start building something. We're definitely anthropomorphizing quite a bit. Who knows what cells really want to do, whether they want to do anything. But they are definitely helping somehow with this self-assembly process. Okay. Over a few hours, you start to get something that looks like this. So this is a lot more cells coming together. This is about 2,000 frog skin cells, and you can definitely see them pulling together into this mass. Most early organisms like to sphericalize. All the spherical cells that make them up, they tend to try and pull together into a sphere of spheres. And then development, all of the genetic signals telling that this mass to build itself into a frog or a human is pushing against this inbuilt tendency for the cells to just want to clump up into a ball. Okay. Once we have this ball and it stabilizes after a few hours, Doug comes back in with his surgical tools, microsurgical tools here, and starts to carve away the unwanted material. You can see on the right, this is a micro-cauterization device. It's a very, very small, very hot wire, and it is burning away some of this excess material. So Doug can either scrape with a very, very small needle or knife the unwanted material or burn it away. You'll notice that he's burning across into this sphere, which when flipped over is going to become the four legs of the Biobot sculpture. Questions? So far, so good? Okay. Last step, you can see he's removed more of this material. You can see these four stubby legs pointing upward, and Doug's going to come in now with micro forceps and very gradually sculpt these final features as best he can to capture the 3D shape of our little soft quadruped that we showed him originally. Okay. That concludes the construction of a Biobot sculpture. What you're going to see on the next slide is not a Biobot sculpture, but the physical Xenobot moving. In order to move, it needs that heart muscle tissue. So what Doug did in that case is more or less exactly what you just saw. But when he was injecting cells into that shallow dish, he would inject a thin layer of skin cells. Then he'd collect heart muscle tissue and eject a small thin layer of heart tissue on top of the skin on top of heart on top of skin, heart, skin, heart, according to our design. So basically building it up like a sandwich. Doug is very talented, but not talented enough to place an individual heart muscle cell where it's needed. So there are some limitations in what he can build, which again, we tried to build in his constraints when we evolved these designs in the first place. So remember, when we cross the sim to real gap, there's kind of two hurdles we need to get over. Can you build it in the first place? And even if you can build it, will it exhibit in reality what the evolutionary algorithm predicted it would do in simulation? Okay, huge amount of buildup. Here we go. So here's the evolved design on top, and the physical Xenobot in the bottom. Our virtual camera in the physics engine is looking kind of from the side. The micro camera that's looking down through the microscope is looking down from above. So not quite the same camera angle. Did we cross the sim to real gap? Kind of. Definitely doesn't have exactly the same shape, right? Obviously, these very sharp edges don't exist in the physical Xenobot. Cells tend not to like sharp edges. They're trying to sphericalize, right? They're trying to come back into a sphere of spheres. You'll notice the Xenobot kind of rocking back and forth and moving forward, more or less. You can't quite see from this video the question that was asked a few minutes ago, which is, are the frog heart muscle tissues synchronizing? Very hard for Doug to test that in reality, to actually see that in reality. We're doing some work on that now to put reporter proteins in so things fluoresce, so we can better see what's happening during behavior. Doug told us, generally speaking, there wasn't much synchronization, which was pretty good because we didn't assume there was any. So our conservative estimate was probably a good idea. In order to really test whether we cross the sim to real gap, however, we need to ask the following question, which is, maybe if you just throw together frog skin and heart tissue, you'll get something that's kind of like a wind-up toy, and it'll just move regardless of how you combine skin and heart, right? So we're trying to be skeptical here and not assume that we cross the sim to real gap because of this evolved design. So we did a control test, which is we took the physical Xenobot and we flipped it on its back. If the Xenobots will just tend to move no matter what distribution or what shape they have, a Xenobot on its back should move just as far forward as the Xenobot on its front. Make sense? Okay. So you're gonna see the same Xenobot but captured from a different camera angle in a moment. And what you're going to see in the first segment is you're going to see the Xenobot right side up. There it is, right side up, sped up quite a bit. And then we're going to flip it on its back. And it doesn't move. Okay, so the 3D shape and the way that heart and skin is distributed is what's conferring motion onto the Xenobot. Questions? Yes?
Where was like the power?
Where's the power? Any biologists here? Where's the motive force coming from? These cells were taken from very, very early frog embryo, basically a frog egg. Where does an egg get its power from? When we were all eggs, where did you get your power from? What's inside an egg that confers power? What's the energy source? If you eat an egg as an adult, you get quite a bit of energy from it. You get quite a bit of power. Where is it coming from?
The protein.
The protein, but where's the protein? Definitely the protein. Where's the protein concentrated in an egg?
The yolk.
The yolk, absolutely. So each individual frog cell and human cells, and every mammal and every higher animal that comes from an egg, the egg, small amounts of the egg yolk are actually incorporated into the cells. So the cells are carrying very small amounts of egg yolk inside. It's the battery. Each cell has its own battery or power source, which is the protein that it cannibalizes from inside the yolk, turns into ATP. ATP powers the heart muscle tissue to beat. Okay, so that's good. No, we don't need any external quote-unquote cables, no umbilical cords. The Xenobot has an onboard power source, which is great. But like any battery, there's a limitation, which is onboard power is great. It's got a limitation, however.
Motor force.
Motor force, so batteries, human batteries tend to be relatively weak. Egg yolk batteries are pretty strong. There's a lot of power packed into an egg yolk. No matter how strong a battery is,
It dies eventually.
it dies eventually. So the Xenobots that you see here, they do their thing. They move for about 7 to 10 days, and then they gradually stop moving, and then they just rot and die away like any other biomass. Okay. Whether they rot or whether they die depends on whether you consider this an organism or not. Okay. Okay, so I showed you anecdotal evidence that we crossed the sim to real gap, but anecdotal evidence is usually not enough. So we did some quantitative testing to show, in general, we could cross the sim to real gap. So Doug built several versions of this design, put them down in the Petri dish, and watched how they moved from an initial position. And that produced the blue-green curves that you see here. So you can see, generally speaking, the blue-green curves tend to move towards the right, which is good. That's what we want them to do. We took this design and we replayed it in VoxCAD about a dozen times. And we got the pink curves that you see here. We replayed the same simulated Xenobot multiple times, and it produced different trajectories. Why do you think that occurred? Same Xenobot, exact same design, produces different trajectories.
It was like the offset was randomized, and so was the amplitude of every part cell.
Yep, not the amplitude, but we randomized the phase offset. Every voxel in every simulated Xenobot has the same frequency, same amplitude, just we randomly set phase offset at the beginning of each simulation, and then hold that phase offset for the remainder of that simulation, which produces these different curves. You'll notice that the blue curves don't lie exactly on top of the pink curve. That would have been 100% sim to real transfer. We were not that hopeful. But generally speaking, the simulated and the physical Xenobots tended to move in the direction we wanted them to move. We counted that as a win. The referees of this paper also agreed with us, and it got published. Okay. As I mentioned, we did this control case. We took all of those simulated and physical Xenobots and turned them on their back. And in both vivo, in reality, and in silico, in the simulation, that also disrupted the desired function. Questions? Okay. When we published this paper, people noticed a lot of public exposure. You make frog bots, you got to expect a little bit of public exposure. You can go and have a look at some of the popular media coverage of the Xenobots. Just in December, me and the Xenobots ended up on the Colbert Show, which was a lot of fun. You can go watch that video again at your leisure. Okay. That was the first Xenobot experiment with individual Xenobots. For fun, since Doug has put all this effort into making these Xenobots, they lived for about 7 to 10 days. Once we got all the sim to real data that we needed that I just showed you, why not put a whole bunch of them together in a dish and see what happens? So Doug, in the bottom, I'm going to ask you to watch the bottom video first. It's pretty difficult to see. You might have to play this on your own laptop. Doug put a few very, very small glass beads in the bottom of the dish, and those beads are smaller than the Xenobots themselves. The Xenobots, just based on the way that they move, they manage to bump into some of these beads and move them around. But they also, when they bumped into one another, they seemed to hold hands for a little while and rotate about their common center of mass and then head off in different directions again. Why? Given the details I've told you so far, why might that happen? They're a little bit sticky. Now, at this point, they've been alive for 3 or 4 or 5 days. The stickiness is going down. They're definitely much less stickier than they were at the beginning. That's part of it. What else?
It's because they still have this motivation to like be multicellular and like come together more.
That's a great question. Our biology colleagues are scratching their heads about that at the moment and trying to determine whether that's true. Do the cells actively want to try and come into contact and remain in contact with other cells, even if those other cells are part of another xenobot? That may be part of the story. We don't know yet. What else causes them to temporarily adhere and rotate about their common center of mass? There's a hint in the top video.
It looks like they kind of just run into each other, and then since they don't have any sensor data, they just keep trying to move that way until they eventually break off.
Absolutely, right? So these are just machines, as far as we know, open loop controls. So they manage to crash into one another. They're not perfect spheres. They do have little indentations and geometric details on their surface. So sometimes they kind of lock together. You can see that a little bit more in the top video, where again, in the top video, these are simulated open loop robots. And if they keep pushing into one another, they'll remain temporarily locked until there's a little bit of agitation and off they move. That was kind of surprising to us, but maybe in retrospect, not that surprising. It suggests new opportunities for extracting collective intelligence from these very, very simple individual robots. Question?
I mean, could it have to do with the fact that there's like the beads and maybe that's the path of least resistance, is kind of like staying together and just moving in the circle?
It could be. So the glass beads are also kind of obstacles. So these Xenobots are moving very lightly over the bottom of the dish. So you can kind of think of these Xenobots as like running on the bottom of a swimming pool. They're in contact with the bottom of the dish, but remember they're also immersed in room temperature pond water. So it's possible that they kind of hit these glass beads and maybe they roll over a little bit, or it alters the trajectory that they're moving. This is all very, very small. It's hard to actually see kind of what's going on here.
Why add the glass beads?
Why add the glass beads? Good question. Why add the glass beads? Why were we interested in that? Unfortunately, there are huge amounts of microplastics in our waterways. Very, very difficult to get them out. If there's a water bottle, you can imagine a robot boat or a drone grabbing it and pulling it out. Microplastics are much, much more difficult to extract from natural waterways. So we're interested in, could these very, very, very simple robots act like Roombas and basically clean up things? Could they push very, very small amounts of microplastics? It's possible that next generation, or maybe the next generation of Xenobots, could be designed to have certain cells that are sticky to microplastics. So as they move about, microplastics just stick to their surface, and when they die and float to the surface, we collect them and off we go. Applications for the Xenobots are still probably years and years off, but we just kind of wanted to see whether we could recapitulate or redo some classic robot experiments, right? The Roomba vacuum cleaner was the first successful commercial autonomous robot used in the home because it has a very simple purpose. It tends to be able to get places that are hard to reach or very difficult or onerous for a human to reach. Same idea here, just on the micro scale. Yeah? Okay. Other things we noticed is that sometimes a Xenobot will seem interested in a particular glass bead in its environment. This is much harder to explain. Is the Xenobot in the bottom seeing and becoming interested in this glass bead? Seems unlikely, because we just mentioned that we didn't build any sensory cells or sense organs into these Xenobots. However, the individual cells themselves have very sophisticated sensory receptors on the surface of the cells, and cells communicate with one another. So it is possible that there are sense, think, act loops going on inside small groups of cells inside a Xenobot. It is not impossible that this Xenobot or parts of its body are sensing and reacting to things in its environment. It's hard to explain the motion you see in this bottom video as purely mechanical. It's possible. Might be that when it bumps into this bead, it kind of rocks up onto its right-hand side, which causes it to turn in a tighter circle for a while. We are still working to rule out some of these purely mechanical explanations for what's happening.
Could you be able to add energy sources into your solution and then have Xenobot last longer?
Yeah, great question. So it is possible to diffuse nutrients into the water, and cells will passively absorb some of those nutrients. It's not very much. You can keep them going for a little bit longer, but not very long. Yeah, good observation. Okay. I mentioned that one of the 100 designs that we evolved is a hollow structure. So why not place something inside the hollow Xenobot, and then as it moves, it transports this small yellow pellet. Why did we bother trying that?
That'd be like a medicine thing, like you mentioned the other day.
So we talked about this last time. Intelligent drug delivery is an emerging application domain in synthetic biology. There are many human diseases and pathologies in which it is very difficult to get very powerful and potentially dangerous medication exactly where it needs to go, right? Best we can do is swallow or inject medication or a therapeutic that then starts to diffuse outward from wherever it ends up, which is often better than the alternative, which is doing nothing, but it's still not a very good thing to do to the human body. Can we get better at more intelligently delivering drugs where they need to go? There are a whole range of technologies that are being developed to test this idea. Could be micro robotics, but I mentioned the human body doesn't like small amounts of metals and plastics. So maybe biobots would be a good potential application or a good vehicle for intelligent drug delivery. Although human bodies don't necessarily like frog cells inside either. In your digestive tract, no problem, but outside the digestive tract, not such a great idea. So where might we go from Xenobots to take this idea one step closer to reality? Can't deliver drugs with a Xenobot.
Could you use human stem cells?
It's possible that this could be done in the not too distant future with human cells. If possible, and again, years in the future and also significant regulatory hurdles for good reason, it may be possible to try and build biobots out of a human patient's own cells, so that if they were to ingest or inject those human biobots, the human immune system would not be triggered because your body would recognize that bot as literally you. We talked really early on in the course about self, non-self discrimination. Here it comes back in a 2020s version. Okay. One last item I want to show you before we switch gears is another potential advantage of biobots over traditional robots is we get to inherit useful function for free. The cells themselves that we're building this material out of, they've had 3.5 billion years in the School of Hard Knocks of dealing with internal surprise and external surprise. Here you see the cells dealing pretty well with internal surprise, which is Doug took one of the Xenobots and cut it almost in half. And already by about 10 minutes, it had started to seal itself back up again. This was not some lucky accident. He injured another Xenobot, and it again sealed itself back up.
Is that why he used the cauterization and burning of the cells?
Absolutely, right? So if you cauterize something, you obviously resist its inbuilt instinct to try and heal itself back up again. Yeah. So in this course, I've shown you torturing of robots 3 times. We saw the evil starfish. You pull off its leg, it's got to adapt its control policy. Last time, we saw soft robots where we lopped off some of their legs, and in some cases, They adapted their control policies. In other cases, they changed their shape to recover. Here's one step even further. Biobots, after injury, tend to return to their default or imprinted shape and carry on with whatever it is they need to do. We didn't need to train or evolve Xenobots to recover from injury. They do it for free because that's a very old and very generally useful adaptive strategy. Okay. So just taking a couple steps back from Xenobots now, this is just one particular example of this larger emerging field of synthetic biology, which lives somewhere at the interface between AI, robotics, and biology. Most of synthetic biology to date has focused on robots, but not AI. So it's a human engineer, a human bioengineer, coming up with a design and building it by combining biological and technological components together. Those are biohybrids. You can go have a look at some videos of some of these. This is about a centimeter-sized jellyfish that combines artificial components like silicone, wherever the soft voxel is, and rat heart muscle tissue, which also expands and contracts and gives you jellyfish motion. Here's another example of a biohybrid. I've got a video of it. You can see it at work here. There's the rat heart muscle tissue contracting and expanding. Here is a biohybrid Bratenberg vehicle. They're flashing a light, and the cells, they've included cells that are sensitive to light. More light falling on the right-hand side of this biohybrid causes the rat heart muscle tissue to contract and expand at a faster rate, causing it to turn away from the light. Yeah, this is the vehicle 2A, the coward. Okay, again, all human designed. So what I just showed you in the case of the Xenobots is, again, in the spirit of this course, trying to take a couple steps back as designers and not assuming the form and function of the machine beforehand, but asking an evolutionary algorithm to come up with that design. In this case, the evolutionary algorithm was allowed to reconfigure tissues to make reconfigurable organisms. There are other biobots on the way where instead of reconfiguring the tissues, the evolutionary algorithm is allowed to manipulate other aspects of the biology to produce new kinds of biobots. We haven't published on that yet, so I'm not allowed to talk about it yet, but stay tuned. Okay. All right, that concludes our discussion about Xenobots. We have 15 minutes left. We've covered a lot of material in this class. We've covered some very new material, some very old material. Questions, comments, things that have been nagging at you that you wanted to discuss? Yes?
Has the Xenobots project received any ethical criticism?
Go to Twitter, type in Xenobots. To say that it's received some negative ethical commentary is an understatement.
What is your defense against it?
I've got plenty of defenses, but rather than you hearing my defenses, let's start with the criticisms. What do you think some of them are?
Well, that last one that you showed us that could carry something.
Carry a potential drug pellet?
Yeah, it could be like a good drug or a really bad one.
Could be a good drug or a bad drug, right? Any technology, going back to stone axes, could be used for good or not so good purposes. Whether or how much responsibility the scientists or the discoverers of a technology have is, again, an ongoing debate that's been going on for centuries. What are some other ethical issues here? The bad guys and girls might use it for nefarious purposes.
If the self-replicating ones are too good,
Absolutely. I don't think we're going to have time today to talk about the self-replicating Xenobots. What we've talked about here, the original Xenobots were published back in January 2020. Got lots of public media attention, lots of criticism and argumentation about whether these should even be pursued. 2 months later, another very small creature showed up on the scene and stole the spotlights away from the Xenobot. We're still battling with that particular creature. The self-replicating Xenobots we published 5 months ago now in December, which triggered a whole other stream of discussion about self-replicating machines, which of course, as Hollywood has told us, the minute you make it, it's going to spread out of control and turn the planet into gray goo. It's known as the gray goo hypothesis. Other criticisms. You're missing the most obvious one or ones. What do you think the first criticism was?
The whole like ripping apart cells and putting them back together.
Absolutely, right? So we are obviously tearing apart early animal embryo and using it for scientific purposes. There is obviously, again, a very old debate on whether or how animals should be used to advance human knowledge. It's another whole other, we could obviously spend whole classes discussing that. What else?
Well, on the animal part, like, is there a reason why you picked the frog?
Absolutely, there's a reason we picked frog, and I mentioned what it is when we first started talking about Xenobots. Why frog?
It was because they did like the whole eye on the back experiment.
So it turns out that frog in particular tends to be relatively accommodating to anatomical reconfiguration. Not just frogs, others as well. Frogs are also relatively low down on the scale of animals that humans care about. Unfortunately, for worse purposes. The biohybrid that I just showed you that was built from rat heart muscle tissue, this one is, in my personal opinion, much more ethically problematic. Extracting rat heart muscle tissue from rats is much more uncomfortable for the rats. I will leave you to go read about it if you're interested in that particular ethical component of this work. Other ethical concerns. There's lots.
These cells aren't just motors, so they could have unexpected things happen.
Unexpected things could happen. We spent a lot of time in this course talking about perverse instantiation. There is even more perverse instantiation possible when you're working with living materials, because the robot components themselves have their own ideas about what they want or don't want to do. Here's a positive example of that. We got additional good functionality for free, but it's hard to say what else we are building into these machines that don't exhibit themselves until after deployment. Yeah, absolutely.
This good functionality, as far as the experiment goes, could be perceived as kind of a scary thing. The fact that it gets damaged and it heals itself can be scary.
Why is it scary?
Then they can't be destroyed.
They can't be destroyed, right? It's harder to shut this off, right? Which again, humans have been discussing for centuries, and Hollywood has had a good time with over the last few decades.
Life finds a way.
Life finds a way, right? The Jurassic Park reference comes up all the time. Jurassic Park comes up all the time. What other movie is always mentioned with these kinds of things? The Terminator, right? You missed the Terminator one. Why would we make Terminators? The first Terminator movie, you pull off all the body parts and the machine just keeps going, right? What else? Other ethical concerns. We're obviously not going to answer any of these ethical concerns. Each one is exceedingly complicated. It's important to think about all the various ways that society might have a problem with this technology if, in the not too distant future, it becomes practically useful. Not there yet, but worthwhile talking about it now.
Like when you get to the point of like using this using it in humans, like we talked about the autoimmune response.
Yep.
Could be an issue.
Yeah, absolutely. So who knows, right? Maybe we can sidestep the immune response, but the devil's in the details. Maybe that's not such a good thing.
Furthering that, when you said that one of the possible solutions is using human stem cells, that a lot of people are not stoked about.
Absolutely. So in this particular country, the fact that we're using stem cells is an additional ethical issue, Not so much of an ethical issue in other countries. This one, absolutely. Yeah.
There's a question of whether the actual Xenobots can feel things and if they're like have feelings.
Absolutely, right? Thinking about thinking and thinking about feeling is misleading. Are these Chinese rooms? Is there anyone at home, right? We didn't obviously build in any nerve tissue yet. There are no pain receptors inside a Xenobot, but nevertheless, is it worthwhile thinking about the subjective experience of these organisms? We tend to, for default organisms, for the frog itself and the mouse itself, the human itself, if we're in the process of expanding our circle of empathy to encompass other animals, should Xenobots be included in that or not? What do you think? How would we know? How would we know whether they experience distress? Maybe not necessarily pain in the way that we sense it. How would we know whether they feel distress or not?
I was gonna say if they avoid like negative stimulus, but I guess that could also just be like a brain bird style.
So what if they avoid negative stimulus? When we talked about does this thing have intelligence or not, we ran aground with that question, and for many of us, the only answer is the answer that Turing gave, which is we'll know it when we see it. There is no way to internally investigate or interrogate a human or an organism or a machine and conclude there's intelligence inside. All we can do is look at what it does and infer from its behavior whether or not it's intelligent. Seems like the best we can do at the moment is come up with an ethical Turing test for these kinds of organisms. If they act as if they are in pain or distress, maybe we should assume that they are. Not, again, like all Turing tests, it's somehow unsatisfying. Maybe that's the best we can do. Other ethical questions here.
As they get more complicated, some people might see this as like even more extreme, like Titanic creating like even like new species of things.
Absolutely. So it turns out there's again nothing that special about frog. We could make biobots out of other cells taken from other organisms, or mix and match cell types taken from different organisms, which would be a chimerobot after the chimera. Seems that may become possible in the not too distant future. Another ethical criticism is, aren't you playing God? Are you allowed to do these sorts of things? What else? We got 5 minutes left, so we're not going to have time to talk about the self-replicating Xenobots. I will just tell you how this works. I showed you, we saw the Xenobots moving around and pushing small glass beads into piles. That led our biology colleagues to ask the following question. What happens if you replace these glass beads with more individual frog cells? You'll get more or less the same thing. These Xenobots will push those individual frog cells into piles. Remember that early frog embryo cells tend to be sticky, so those piles will tend to cohere or stick together into a ball. And after a few days, under the right conditions, that pile will start to move on its own. That pile is not moving using heart muscle tissue. Turns out that under certain conditions, frog cells will grow cilia. These are very small hairs on the surface of the cell. You also have cilia in your trachea. It helps you cough out irritants in your airways. They're sort of things that on a normal frog will push contaminants or irritants off the surface of the frog's skin. If you make a ball of frog skin, those frog skin cells will grow cilia, and they will end up beating like oars. And if that pile is big enough, it will actually start to move because there'll be enough of these cilia. So you get what's known as kinematic self-replication. Kinematics meaning motion. So the Xenobots don't become pregnant and grow a baby Xenobot inside and give birth like other animals do. They move about kinetically, and they end up pushing these cells into piles. The piles start moving. You can think of these moving piles as child Xenobots. If you sprinkle more cells into the dish, those child Xenobots will push cells into piles, producing grandchild Xenobots, and off you go. Are we in danger of the gray goo hypothesis because of self-replicating frogbots? Hopefully not.
It always requires our own outside resources.
Remember our discussion about the Penrose tiles when we saw those wooden tiles? They were picky eaters. The self-replicating Xenobots were designed to be a test bed for testing the conditions under which biology will spiral out of control and the cases under which we can stamp something out or suppress or guide replication. That's the goal. That was the goal of the replicating Xenobots project. 2 minutes left. Any last burning questions before we conclude? Yes?
How do the cells that you sprinkled in not attach to the already created Xenobots?
Yeah, great question. So remember that the quote-unquote older Xenobots, as the cells mature, they become less and less sticky. So the stickier younger cells tend to stick to each other. They stick a little bit to the parent Xenobots, but not very much. Yeah, good question. Okay, you have one final quiz due tonight. We will be back here Tuesday morning at 7:30 to see all the wonderful creatures you have managed to evolve over the last couple of weeks. Good luck with the last week of semester and exams. See you next Tuesday.